Good evening, everybody. How's everybody doing? All right, please pardon the technical difficulties and please pardon the delay, but we are now about to begin our Faculty Pub Night October edition. My name is Ray Andrade. I am one of the 3 members of the Library Outreach Department. So on behalf of the entire library team, welcome to Faculty Pub Night. So for tonight's event, we're going to feature Dr. Ben Fitzpatrick, Professor of Mathematics, and he'll be discussing his recent publication, Getting Personal with the Reproducibility Crisis, Interviews in the Animal Research Community. But before we formally begin the program, there are a handful of things that I did want to mention about Faculty Pub Night. Number one, especially this goes out to the students in the room, it is Faculty Pub Night, but you do not have to be 21 to attend a Faculty Pub Night. We do check IDs, so my boy Eric, the bartender, is in the house checking IDs, making sure you're not going to be slick to get some drinks going on. But otherwise the format is chill. At any point, you're welcome to go to get some food, get some drinks at your leisure. Okay? Secondly, it's called Faculty Pub Night, but it is not for faculty only, so students, I understand some of you are getting extra credit. Thank you for coming out. For any future Faculty Pub Nights, please feel free to invite some friends. The library would really appreciate that as well. Secondly, feedback forms. You might have noticed these on your chairs. So a couple of important things. Obviously, please let us know your thoughts for today's events. Also, at the very bottom of the form, if you include your email address to get signed up for our electronic newsletter, you would automatically be entered into a raffle for a $100 Amazon gift card. So hopefully that'll come in handy with those expensive textbooks. All right. Lastly, the library has a fun new initiative. Every month we're going to be printing 50 buttons. So if you want yours today, there's going to be some next to the box for the feedback forms. So for the month of October, this month's button says, if it happens in the library, it's research. Okay? Students, I'm going to repeat that. If it happens in the library, it's research. All right? So anyway, pick up a button and some bookmarks and some stickers. So now we get to formally begin the program. Actually, one more thing, Q&A. So instead of holding questions to the end, Professor Ben Fitzpatrick is welcoming questions at any point during tonight's program. So please feel free. And now, lastly, to formally begin tonight's program, I would like to welcome Dr. Patrick Shanahan, Professor and Chair of LMU's Mathematics Department. Dr. Shanahan.
I only have about 40 minutes here. So
want me to move the slides for you?
Yeah, exactly. I don't think I need this. Thank you all for attending. I really appreciate it. As I said, my name is Patrick Shanahan. I'm chair of the Mathematics Department. I'm pleased to introduce today's speaker, Professor Ben Fitzpatrick. Ben joined the LMU faculty in 2002, and he is the endowed, Father Wallen endowed professor in mathematics. He came to LMU with a wealth of experience both in academia as well as in industry. And so given that background, it's not surprising that his scholarship includes both basic research and applied mathematics. You should think about that term for a second, basic research and applied mathematics, as well as collaborative work that applies mathematical and statistical methods to solve problems in a variety of areas, including business, economics, engineering, the life sciences, and social sciences. He's going to speak with us tonight about his recent publication, Getting Personal with the Reproducibility Crisis, in which he discusses a current and very serious problem in science of the inability for many studies to reproduce their results. So please join me in welcoming Dr. Ben Fitzpatrick.
Yeah, you might need it. The main reason why we ask people to use the microphone is because the event's being recorded.
Oh, good. I didn't want anything on record.
Okay. So thanks, everybody, for coming. Thanks, Pat, for that nice introduction and offer to cover the 40 minutes of slides for me. So I do need to say this is joint work with a couple of people. Dr. Elena Kustova, who's program manager at the National Institute on Drug Abuse, and Dr. Yun Wong, who's a mathematician engineer, works down the road at a small company, Tempest Technologies. So the story that I'm going to tell tonight goes the following way, that there were lots of sort of detection points, I guess, of the crisis of reproducibility, but the one that sort of drove my involvement had to do with pharmaceutical companies' frustration with the published literature. And that frustration became sufficiently well known that the National Institutes of Health decided to get involved. And that leads to the point of the story where we embark on a journey of discovery and people tell us all of their problems. Now, as this is going on in the life sciences and social sciences community, the statisticians kind of woke up and got pretty excited and came up with a bunch of stuff, and I'll mention a little bit of that stuff too. But to sort of spoil the story here, it's not really like, say, The Hobbit or The Lord of the Rings. It's more like one of those French movies like Cachet, where you're driving home and you're still trying to figure out what it was really about. So we're confused about important things. Maybe we're a little less confused, but the things are more important. I don't know. So for those of you who don't really know a whole lot about how drug discovery works, I wanted to talk a little bit about that particular process. And it has multiple sorts of phases. It's really hard for me not to just walk over here and point, but I can't leave the microphone behind, I guess. Let me see if I can take it with me without too much grief. So the process of drug discovery starts usually in labs like the Life Science Building not too far away, where researchers are pursuing their crazy ideas and trying to turn them into gold, or at least publications. And so you'll have many, many, many discoveries in a very early stage in what we would call the preclinical and drug discovery area. Now, when something looks like it's got enough promise and has been tested in certain ways, it goes into clinical trials where it moves from studies in cells and studies in animals to studies in human beings. Okay? And in the phase 1 of a clinical trial is a small number of volunteers who are healthy. And the basic problem of the phase 1 trial is this medicine actually going to make people sick? So they test it on healthy people for safety first, and if it looks like it's not going to have like thumbs growing out of your head or something like that, then you move on to phase 2 where they actually give it to some sick people and see if it has a therapeutic effect. And this goes on for a few years and may have a few hundred patients. And then if this looks promising, they'll go to phase 3 where they have thousands of patients. And for big problems like cardiovascular disease, you may have studies that have tens of thousands of patients run at hospitals all across the country. So in this early stage, there's a lot of lab work, and then there's some work that's maybe a little bit more toxicology, but on the scale of animal studies. Once it passes into the human beings, it gets a lot more regulation, and the Food and Drug Administration is involved. Over here, this is basic research for the most part, and people are, sort of on their own recognizance, so to speak. All right, let's see. I still haven't got the technology down here. So the problem seems to be, at least according to pharmaceutical companies, that this is getting harder and harder. So R&D costs are going up, and discoveries are going down, down, down. And you might actually think that the basic research that's funded by NIH is really kind of the foundation for what the drug companies do, but drug companies, in fact, spend more money on basic research than NIH does. You can argue, certainly, about the ethical behavior of drug companies. I can't personally say I think a whole lot of them, but nonetheless, they're spending. So after a lot of years of somehow not being able to get things to jive with the published literature, so some researchers at Bayer actually undertook a quite rigorous reproduction study. And they picked 76 studies that were published in the literature that they thought might actually lead to something they could sell. And out of those 76 studies, they were able to get the same results that was published in 14 of them. It's not a particularly good rate of reproduction. And they at least thought a little bit about, well, maybe it's the journal. I mean, they started with pretty high impact journals, but they didn't find that the reproducibility was in any way associated with the impact factor or sort of the complexity of the lab. So they were really at a loss. And in fact, they abandoned a lot of lines of research after doing this study, and are, in some sense, trying to figure out what to do. In some sense, maybe they've moved to lobbying and they're trying to get the FDA and the NIH to oversee these things. Now, about a year later, Amgen published a similar study with even worse results. Only 6 out of 53 came out basically with the same results that were published. Now, that's not to say the rest of them were total disasters. In some cases, it was kind of a partial reconstruction, but it certainly wasn't enough to feel comfortable investing in the next level of research activity at the company. Now, I talked to one of the authors of this thing, and I'll say some more about that later on, and I mean, they went to some trouble to do this. They brought people to Amgen who published these papers. They went to the labs of people who published these papers to try to make sure they were doing things as well as they could do. And they do have a vested interest. They are trying to make money. They don't want to, like, show up academics. They want academics to do their work for them to the extent that it's possible. So they're actually, sort of rooting for these things to work out. So here's the question, right? Here's what we see in the publication in Nature and Science, and this is what sort of came out in the lab, right? I don't know if you guys know this movie. It's not very good, but as you might guess from this picture, right? I mean, although Michael Keaton is now an Academy Award winner, I guess he looks back on this the way Tom Hanks looks back on some of his early movies. Now, I should also say the study at Amgen and Bayer, we're a little bit on the small sample side of 50 or 60 studies. There are other sort of replication studies that look at much larger sample sizes. And while it does improve a lot, it's like a coin toss. You pick up a paper in Science or Nature, there's a 50-50 chance you got something that's worth looking at, right? Now, the Friedman, Cogburn, and Simcoe paper, those guys are economists, and so they sort of did a lot of analysis of the sort of methods and costs and stuff, and they come up with something like $28 billion wasted. Bazoo and Chalmers in Lancet, I mean, you see these places where these replication studies are getting published are also pretty high-impact journals. Estimate worldwide $200 billion is being wasted on bad research. I mean, again, we'll sort of see some arguments about that. Now, the economics paper goes a little bit further and tries to dive into what the problems might be in the papers, okay? And they separated it out into these 4 categories, the 4 horsemen of the science apocalypse, I guess. And while we see the lion's share would appear to be sort of maybe not well curated story on what your materials were, roughly half of it could be associated with people in my business in statistics, study design, and data analysis and reporting. So within the study design, people talk about low statistical power for the detection of true positives. They found inappropriate methodologies being applied. One of the biggest problems is what do you do with weird data? So you're excluding subjects, you're removing outliers. How do you define that? What does that mean? Okay. In the analysis and reporting, well, there's an incomplete discussion. Those of you who've written papers for high-impact journals know that there's a real pressure to squeeze a lot of stuff into a very small piece of real estate. It's not uncommon for journals like Science to maybe have like a 2,000-word limit. you can't really say a lot. And there's no payoff for writing a supplemental. I mean, they may require it, but still. Explaining these things in enough detail where somebody could actually replicate it becomes really tough. So, after these papers came out, and they start to show up on the evening news, and The Economist writes about trouble in the lab, and you see all these kind of horror stories on NPR and that kind of stuff. The NIH got excited. Of course, what do? They have a workshop, and they bring in a bunch of geniuses at well-funded laboratories, and they come up with a list of things to do. And it's basically what scientists sort of think they're doing anyway, right? Things about reporting, things about recording, lab protocols, blah, blah, blah. But NIH got motivated to actually add some requirements to proposals. So the scientist who wants money from the NIH has to come forward and say something specific about the scientific premise on which this thing is based. What's the prior research? How does that lead to what I want to do? Well, you would sort of think that was already going on, but I don't know. Now there's some additional requirement. Rigor in experimental design, justifying the number of subjects, explaining what analysis you're going to do and why. Another one that's kind of a topic in its own right is sex as a biological variable. It's remarkable how much medical science is based on the behavior of males. Male rats, male mice, male human beings. And I don't know, we should be checking that out too. So here's sort of how the story unfolds, right? We have these companies saying this is a bunch of crap. We have NIH saying we need to do something about it, and we have the academic community saying we know what we're doing, get out of our face. So that all leads me up to actually what I was going to talk about. Gosh, I've been going for 20 minutes here, and I haven't even got to what I was going to talk about. There's an old statement about stories, and I've seen it credited to Dostoevsky, but I'm not sure about that. It says there are really only two plot lines in all of fiction. One is a stranger comes to town, and the other is somebody embarks on a journey. And in this case, actually, the story has both elements because a stranger came to town. So the people at NIDA actually approached this small company because of, in some sense, their technical capabilities, and said, hey, we have this need for something, you should write a proposal. And so we got together with them and wrote a proposal, and I thought, oh, it's going to be great, I can scratch my gray beard and give grandfatherly advice about doing calculations. But after they gave us the money and told us to get to work, then they also said, we want you to go figure out whether or not this is really needed. But you thought it was needed, you gave us the money. But no, you need to go talk to people and find out if they're going to buy it. So I should say, this is not a basic research grant. This is a small business grant, which they intend for you to develop a product and sell it and make money. Okay? And so I'm consulting with small business to try to help them get something that works and figure out how to sell it. So they asked us to actually go out and talk to people. They gave us about a month of lead time to figure out how to do this, and then they gave us 6 weeks to do it. All right? So we're spamming, let's see, I think I probably have a slide about that. We're spamming everybody trying to figure out how to do this, but we thought, this is going to be a breeze. Everybody seems to know this is a problem. We're going to do this. It's going to work great. People know we have these problems with false positives, with false negatives, and I find it strange that I'm wistful for a time 10 years ago when we were asking, is our children learning? By today's standards, I don't know. So we begged, we spammed, we called, and we somehow suckered about 130 people over a 6-week period into talking to us. Okay. Now, the majority of those people were faculty members. The business people worked for device makers, for pharma. They're trying to make money and they don't want to spend a bunch of time. Faculty members like me love to sort of show off how much they know about something, hence faculty pub night. But faculty members were full of stories about reproducibility disasters from other people. Okay, we talked to a few people who are at private foundations that are like focused on Parkinson's disease or Huntington's, various kinds of specialized things. We talked to a few biostatisticians. We talked to a bunch of government people, both at the FDA and NIH. And we talked to lots of graduate students and faculty members as well as postdocs. IACUC is a committee that manages animal welfare. So every university that has animal researchers is going to have one of these committees, and they have to oversee and make sure animals are well cared for and not abused. We ran all over the place for those 6 weeks, driving all over Los Angeles, going up and down the coast, going to meetings across the country. But before I get into that, I do need to sort of set out a little bit about the statistics mumbo jumbo. So this is a little bit of a side trip from the journey where you have to pull over and look at the map, I guess. Okay, look at the map, I guess. You have to look at your phone. Nobody in here knows what a Thomas's guide is, I guess. So there's the truth and there's what you can prove, right? If you ever watched an episode of Law and Order, you know this. And we talk a lot about type 1 errors or false positives and type 2 errors, false negatives. So when you think about a type 1 error, you want to think about somebody who's convicted incorrectly. They were really innocent and they were found guilty. Something is discovered when nothing is there. Okay. The flip side is the guilty person goes free. There's some technicality or what have you. You didn't quite have enough evidence. Seems we sort of experience that on a daily basis nowadays. I'll let you guys think about that. True positive is what we're after. Okay? True positive is what we're after. And the currency in this business, outside of statistics departments, who generally really frown on this particular measure of merit, is the p-value. Okay? And the p-value is some kind of index that you use to assess whether or not you believe you found something. And the smaller the p-value, you feel like the stronger the evidence. I could give a whole, and my students know that I have given many, many lectures on this kind of stuff, and they're still, some of them at least are still here. But what the p-value really tells you is the following kind of weird thing. If you can actually replicate the experiment under the known conditions that nothing is going on, this is the fraction of times you would actually think you found something. Even if you sit and think about that for a few minutes, this is like some weird French movie. Okay, it's a little bit kind of, it's like scratching your ear like this instead of like that, you know. But it's sort of it's a currency that people feel like they understand and use. Now, where does this come from? So Ronald Fisher, the famous biostatistician, actually sort of plopped this down and said, 1 out of 20 seems pretty reasonable to me. 5% sort of corresponds to plus or minus 2 on a standardized scale. So this ought to kind of work okay. All right? So let me remind you some other sort of relevant quotes. 640K is more memory than anyone will ever need. That one you can actually track down. There's another one from Thomas Watson, who was the chairman of IBM, who said, I think there will be a market for maybe 5 computers worldwide. Now, that's one of those kind of quotes that it's not clear he really said that. But the chairman of the company that built the Titanic also said there's no danger that the Titanic will sink. So I don't know about Sir Ronald Fisher's quote here as to whether or not it's really in these two categories, but maybe so. But if we think about what we just learned about reproducibility, so we're saying we would expect, as science operates and produces positives, that's basically the only thing that gets published is a positive result. The false positive rate should be 5%. So we might look at the literature and think, well, 5% of these might be bogus. I don't know, is that the right calculation? I'll let you guys think about that. All right. If we see a p-value below 5%, then we write it up, submit it, and hope it gets published. But what we're seeing here is a false positive rate of something like 50%. Is that, but are those the same things? Okay, that's the math lecture. I could give down at U-Haul. Okay. This is no equations. Okay, 2 inequalities, but that's not so bad, right? No equations the whole time. Okay. No proofs. I don't know. The math department might kick me out after this talk. So back to the journey. Okay. Here's what we really found. Okay. People sort of argue about what you really mean by the term reproducibility. Okay. And so we had to sort of, every time we talked to people, come up with some agreement about what that was. Now, that said, there are plenty of surveys. I mean, Nature published a survey of like 1,500 scientists, and everybody seems to believe there's a reproducibility problem, even if they don't know what it means. Academic culture is a big problem. Okay? Biomedical experiments are quite complex and getting worse. When you're in a complicated study, well, if you start thinking about what you might wish you had studied instead of what you started out to study, you can get in this kind of weird feedback loop of chasing a result. Okay? The layout, although by the time you've published it, it's too late, the layout, the experimental design, is also quite a big issue. And in fact, beyond the sort of what academics will complain about, the culture of academic life, the statistical stuff is often sort of believed as sort of the second largest problem. And many biomedical researchers are quite uncomfortable with statistics, but that certainly doesn't stop them from doing it. So the ACM, the computer science group, has a sort of taxonomy of reproducibility things. So at one sort of fine level is you do exactly the same thing and get the same result, okay, with the same setup, okay. Then you invite somebody else to your setup and do the same thing and get the same stuff. That's repeatability in the computer science lingo. And reproducibility is actually, you have a different team, they set it up their way, in some sense according to your spec, but their way, and if they get the same thing, that's reproducibility. Now, Goodman's a statistician. Finelli and Ioannidis are biology people. Ioannidis is a medical doctor up at Stanford who has written a lot of papers with the title, "Why All Research in X is False." I didn't know you could make a living doing that, or I would have tried that earlier. Seriously, Ioannidis got zillions of papers like that. Okay, and they define various levels of reproducibility. Okay, can you follow the processes and the protocols? Can you get the same data if you do that? Having collected that data, would you make the same conclusions? You might think that's a foregone observation, but not necessarily. Then there's some of these sort of generalizability and robustness things. Under the same, if you tweak it a little bit, do you get the same thing? If you tweak it a little bit more, do you get the same thing? Okay? Now, it's a major issue, even at these sort of further from the state of the experiment levels of reproducibility, because the translation from cells to animal models to human beings, those are big steps. Those are very big steps. Now, so here's a couple of stories, and I say that hold up because we heard things like this from many researchers. Okay? So we talked to somebody who'd moved from Stanford to Berkeley from a postdoc job to an assistant professor job. And the first thing they tried to do was replicate their last paper in their new lab. And they literally moved the stuff. They didn't buy a bunch of new equipment. They literally moved everything they had to a new space. And presumably the only thing that was different was the space. And perhaps how they had to lay things out because the space wasn't identical. And the person actually said, I must have repeated this 100 times. I don't know if I believe that because it's pretty expensive to repeat experiments. But could never recover and never knew why.
Was it the water?
I don't know.
Girls here, we both had this experience where it's the water, like literally.
So another story that's similar to this, Heather told me 3 years ago, was she had a friend that they'd done a study, and they were trying to replicate somebody else's results that looked kind of interesting and related to what they were doing. They couldn't get a replication. They were on the phone back and forth. They traveled to that lab. Those guys traveled to this lab. And in the end, after lots of failed attempts at squaring things away, they decided the problem was, in lab number 1, the animals were living in cages that were papered, if you will, at the bottom, with wood chips. And in the other lab, they were papered in the bottom with corn cobs. Now, could that really be the difference? That was the only thing they could come up with that was different about the two studies. So, the fellow from Amgen told me this story, that one of the people that they'd been trying to replicate, he met him over coffee, and he said, you know, we did this over and over and over again. We must have done this 50 times. Again, in this case, I might believe it because Amgen was pumping an enormous amount of money to try to figure out what the problem was. And the study author actually said the following thing. We repeated it 6 times. One of them worked. That's what we published. Okay?
Does not seem very
This seems very unethical. Okay? So, and while we're on this topic, here's something. If you see a study that has a very weird number of subjects in it, that's an animal study. Now, survey research, you get what you get. You get 419 people, you get 131 people. When you're asking for volunteers, you get what you get. But when you're doing an animal study, you pick a number of animals. And it's probably like the number of cages you've got. Or the number of slots in your centrifuge. Or something like that, right? But if you see a study where group 1 has 21 animals and group 2 has 23 animals, you got to ask yourself why. And if they don't say, these test subjects died because of X, or these test subjects were excluded because of Y, you got to really wonder. Okay, so a journal editor told me the story. They had this paper that was published, that was submitted for publication, and had a p-value of 0.047, which is like just under the wire, just a little bit less than 5%. And it had 21 animals in one group and 23 animals in another group. Okay? And the discussion about what the statistical methods were was a little bit vague. And so in the back and forth of trying to figure out whether or not they wanted to accept this and the referee's complaints, they found out the following thing. They actually started with 20 subjects in each group, and the p-value was like 0.056. So they deleted a subject that looked a little bit weird, and the p-value got a little bit closer to 5%. So they added a couple of subjects and got it under the wire. Okay? Now, my students have in class seen the capability of doing this with some small tweaks of numbers. You can change the study from being not publishable to publishable with some small tweaks. Okay? These are issues, and so that leads to this problem of what is it in the academic culture that makes people do this stuff? Okay, the pressure to publish, especially if you're at Stanford or Berkeley or UCSF, is enormous. Okay, and the only thing you can publish is something that comes out with a positive result. Negative results are really hard to publish. Okay, high impact publications are a must, okay? A guy who's director of a cancer center at a really respected university said the following thing. If an applicant for a job doesn't have a publication in Science or Nature, they got no hope. Even though we might know that the Journal of Immunology, for example, is at least as high a quality, in fact, probably a better publication because it's going to discuss the problem in more depth. But this is like p-values of 5%. This is the currency of the realm, okay? High impact publications are a must, and things like Science and Nature, these are very terse articles. It's very hard to explain what you did, okay? Now, other things that happen involve grants, and if you go to a medical school like UC San Diego, you'll find there's a lot of the faculty that get zero salary from the university. All their salary comes from the grant money they raise, right? Now, in mathematics departments, that's almost never the case, right? In statistics departments, you might see some of that, and people work in like academic consulting centers, but grants are an enormous problem for people doing biomedical research. And you get somebody that's got some really large-scale, multi-million-dollar thing going on, this person is like the CEO of an operation with 20 or 30 employees. They're not overseeing that. They probably have a weekly meeting to find out what's going on. Everybody gives a 5-minute elevator pitch, and you know the inmates are running the asylum, so to speak, right? The lab of Dr. Tar and Professor Feather. Now, if you talk to academic people about industry, many of them are quite disparaging, right? Those people couldn't hack it in academics, and so they went into industry. And so these industry replication studies, they just couldn't do it, okay? The problem with that is, you know, I mean, Bayer, Pfizer, these companies are trying to make money. And they can't make money selling sugar pills. They can't come up with a bunch of bogus medicines and sell them. I mean, you have to do that on some off-brand cable all-access channel, right? I mean, you know, some vitamin supplement guy can do that, but real pharmaceutical companies are not going to do it, okay? Negative results also have different sort of interpretations. To an academic, that's a failure. Toss that in the drawer. We talk about the file drawer problem as having all these actual pieces of data about things that never get found out. Industry is a lot more sort of tolerant of this stuff because they really want to know what works and what doesn't, what has hope, what doesn't. In some sense, both of these are sad because if you really have a false negative, you've missed an opportunity, okay? Industry, it turns out, actually has a lot of underground collaboration, which if the management knew, they would probably be very unhappy. But some poor schmo working in a lab is trying to replicate something, get something to work, and it doesn't work. Well, so they call some guy they went to graduate school with or some former colleague and say, you know, I tried this, it didn't work. And they find out, well, that company is also working on this thing. And they also failed. And that's actually what led to these Bayer and Amgen type of studies. Industry has like a very negative view of the literature in biological and life science, a very negative view. Industry people uniformly said, oh, 95% of that stuff is just crap. I mean, we think, you know, peer-reviewed literature, that's where it's at, but I mean, think about an expensive lab study. What does a peer review do? In mathematics, peer review is pretty easy. There's proof, you check it. I believe it, I don't believe it. If you read a science paper and, you know, they've got this $500,000 instrument that you couldn't hope to get time on, how would you even sort of imagine how that works, okay? Industry also has multiple thresholds rather than just publish or not publish. In early phases, they might be much more willing to tolerate false positives because they don't want to miss possible positives, right? They don't want false negatives in the early stages. But as things progress, they're going to tighten those thresholds. And eventually the FDA gets involved, and they really hate false positives. They don't want to be approving a medicine that people are going to buy that is ineffective. that's going to blow back on everybody and create a lot of unhappiness. Okay? So what's the solution here? Okay? Academics almost uniformly say, well, you know, quite frankly, this is the process of science. People do things, it's not quite right, we eventually figure it out. I would remind you that the Amgen slide had the number of times these bogus papers, well, non-reproducible papers, got cited. And some of these papers live for years and years and years and have hundreds and hundreds of citations. And they're basically non-reproducible. So academics don't want really anything to change. And even the new NIH requirements that you work a little bit harder on your proposal are, you know, pretty tough and resistant. Now, industry says, look, we can't tolerate bad results. I mean, if we publish something that's non-reproducible, what that means is, you know, we've released a medicine that causes problems and we're going to get sued. And it's a totally different ballgame for like writing a paper and maybe you get caught and you have to write some kind of retraction. And the FDA, once it gets to sort of the human trials thing, the FDA is on these guys like glue. Okay? They see there's no consequences in academics for these irreproducible publications, and what the NIH should do is create an IRS and audit the labs and punish people who are not doing things correctly. I don't know, you guys can argue about that. Assessment at this place seems quite burdensome as it is. I don't know what it would do to have labs audited. Okay? Lord knows how bad I'm going over time here. So biological systems are complex, and I would say that social science is harder than biology, and biology is harder than physics, and it's actually for the exact reason that most people think it goes in the opposite direction. And that is that physics is modelable in a very highly predictable manner with mathematical methods. That you can argue about how gravity works and make very precise predictions so that you could build something to land it on the moon without having to throw 50 things up there and see if they hit. Right? Physics doesn't work that way. We don't just like drop a bunch of things and see what happens to them. We sit down and do some calculus. The unit of analysis in biology is so much more complicated than a chunk of aluminum that it's really hard to mathematize. It's really hard to model in a predictable manner. And we're stuck with poking a bunch of people with needles and injecting things into them and seeing what works. If you've ever had an allergy test, like you don't know what you're allergic to, and they'll inject you with about 40 things and you get all these little blisters and they see which one's the worst one. Oh, you're allergic to this. I mean, that's the way it works. Okay? Now, the pace of technological innovation in life science has been enormous in recent years, and instruments get much more complicated. Running a lab requires all kinds of expertise and all kinds of technical things. You just about have to be an electrical engineer to run a lot of the instruments in a biology lab. The data is very complex now, okay? And if there's this really expensive instrument that like 1 or 2 labs have, maybe Berkeley got one and MIT got one, and those people are cranking out results left and right, how could you even hope to verify that those things are reproducible? I mean, they're almost by definition not. Now, here's where things can get a little bit weird, which is you propose this study and you collect a bunch of data, and now it gets worse because you can collect so much data. And you see, well, the thing I started out with doesn't seem like it's working, but man, I got all this other data over here. Let me try that. And eventually I can fiddle around and find something that's going to test positive, and I get something to publish out of it. Okay? Now, consulting biostatisticians that work at universities across this country will sort of tell you that roughly 90% of their work is, my p-value is bigger than 5%, help me. Give me something I can publish from this data. Okay? And Fisher actually said to consult with a statistician after you've collected the data is probably an exercise in figuring out what the experiment died of. So this kind of p-hack, I would recommend if you can tolerate the crude language, because it is really quite crude. It's not something I could show to a class, but I wish I could sort of bleep out things so I could show it to a class, because John Oliver's take on scientific research and p-hacking is very worthwhile if you can tolerate the crudeness. So we're almost there, actually. That's not too bad. When I see this slide, it reminds me I'm almost done, right? So the arc of the story, we're just about to the climax here. So, and in honor of a former president, is our experiment's design.
15 minutes. You're good on time.
Oh, really? Okay. Holy cow. Strap it on, people. Make sure your tray tables and seat backs are in their full upright and locked positions. So one of the things that is recommended in experimental research is not really statistics per se, but in some sense, the statistical analysis depends in subtle ways on this, is that the people who are doing the work don't influence the outcome of the experiment. So if you know which animals are in a treatment group and which animals in a control group, you might treat them differently. Oh, this poor little thing got a percussive shock. He's had traumatic brain injury. I mean, people have to do research on animals that's unpleasant. You might, oh, this poor little guy, let me, And then when the observations are of a behavioral nature, how is the animal doing? And it's not really like a quantitative instrument cranking out blood pressure or how long it takes to complete a maze. It can be kind of easy to manipulate those assessments. And I'm not suggesting this is like scientific fraud or something. It's just kind of a natural human behavior. Now, another thing that statistics depends on is that the subjects are randomly assigned and are in some sense a representative sample of the population you want to study. If you get a shipment of animals from Jackson Labs or Charles River and they're all in this cage and you start grabbing them out, well, the first ones you grab out are probably the ones that are excited to get out, right? And the last ones you grab out are probably scared. And if it's something that's a study on behavior, you may very well have biased your sampling by not randomizing those subjects, okay? I don't know if you've ever been to, a shelter, like pick up a rescue animal, but you'll see all kinds of animal personalities in those shelters. And the same is true of animals that are coming from an animal supplier. Now, we found in talking to researchers that generally speaking, when they consulted a statistician at the planning stage, it was to get a couple of lines of text to put in their proposal, and that was the end of it. Okay? It was really a formality that they viewed as irrelevant to the study. Okay? So, underpowered studies. NIH will typically accept a type 2 error rate, a false negative rate of 20%, which seems like a lot. Okay? Now, many of the people we spoke to don't even include this kind of analysis or justification in their proposals. But now they're kind of freaking out about whether or not they can figure out how to do it. If you were to think about a 20% type 2 error rate, and you did 3 replications, you got about 50-50 odds of all of them actually replicate versus at least one of them doesn't. This is kind of a high number here. Whoops. What's the wrong button? Okay. But if we haven't sorted out what we're trying to detect, it actually could be worse. And there are researchers who have tried to look large scale at a bunch of published studies. Estimates from neuroscience range from a type 2 error rate of about 70 to about 90%. And those are pretty high. These are what one would consider really underpowered studies. And there are reasons. It's expensive to do these things. You get a bunch of animals, you got to do a bunch of stuff to them. You can't afford cages for 200 animals. You haven't got the lab space. Look at the lab space we have. So here we are at the crisis point, right? Here we are at the crisis point. in order to do this calculation, there's this kind of fundamental quantity that you have to have an estimate of. And it's usually referred to as the minimum scientifically relevant effect size, sometimes clinically relevant effect size. And it's basically, you got to know what would matter to the science. Okay, think about it from a drug perspective. My students have heard this example until they're falling asleep. So I need to look at them, make sure their eyes are open. Imagine an aspirin pill, on average, makes headache go away after a couple hours. Okay? Now, you've come up with a medicine, and it makes a headache go away after an hour and 55 minutes. And it's going to cost 10 times what an aspirin costs. Is anybody going to buy that?
We actually reviewed that paper in Journal Club in my seminar, literally. Like, it was the difference between, like, making the headache go away in, like, half an hour versus, like, 45 minutes or something like that.
There's an amount of difference that it would matter. And from a practical point of view, different people are going to have different thresholds for this. If you have really severe headaches, you know, even 10 minutes might be worth paying the money. But you need to at least have some sense of it. You know, if it was an hour versus 2 hours, there's probably some merit in that. If it's an hour versus an hour 50, there's probably some merit in that. If it's 30 minutes versus 29 minutes, I don't know, right? But that's not a statistics question, it's a science question, okay? Scientists will estimate an effect size from a pilot study, but that's only an estimate. And that's an estimate of what they think they can detect with a p-value, not an estimate of what they really think would matter. This is a much easier question for pharmaceutical companies or people who are doing applied stuff, like trying to figure out which advertisement brings in more customers, because they can really sort of associate costs with their error rates. In basic science, much harder to assess this, okay? But we did find that, you know, there were a lot of people who said, yeah, we could use some help with this. So, in statistics, we talk about exploratory research and confirmatory research, and we generally mean when you're doing confirmatory research, you really have a question, and it's a yes or no question. And you can say, yes, I found something, no, I didn't, and there's not a lot of leeway. Generally speaking, when you're doing exploratory research, you're trying to figure out a little bit about how the system works, not make some declarative statement, it works the way I think it works, okay? Now, one could argue, I mean, is basic science really an appropriate consumer of statistical hypothesis testing? I think the jury is sort of out on that, okay? But right now, it is the way people make decisions. Bright lines are really tricky, and these cutoffs really create a lot of shenanigans. Every faculty member in here knows exactly what I'm talking about. They've had a student in their office saying, my grade is 88.7, can't I get an A? Because the boundary is like 90%, right? But no, really it's 89.5% because you got to round up. And people would chase these kind of hard lines, right? I mean, a journal has to decide, do I publish the paper or not? The FDA has to decide, are we going to allow this company to sell this thing or not? There are places in the world where there are yes-no decisions, and they require some kind of YesNo Index. But when you think about the difference between the FDA approving, 50, 40 new things every year, 59 was a record-breaking year for the FDA. They were like putting out press releases about that. And with a maybe 13, 15% success rate, there's not a lot of incoming stuff to begin with. I mean, journal publications, totally different matter. Millions of papers published on an annual basis. Are these things the same? In some sense, they both involve a yes-no decision, but, is a 5% threshold the same for both? I don't know. So, statisticians to the rescue, okay? About a year after we'd gotten into this project, the American Statistical Association had a symposium on the crisis of reproducibility and the disaster of 5% false positive rate. And a year and a half later, they published a whole volume of their premier journal, and it had 40 so papers in it, one of which was entitled Abandoned Statistical Significance. And currently, it is the position of the American Statistical Association that the phrase statistical significance should go away, and people should stop using it. I'll let that sink in for those of you who've written that in papers before.
And replace it with what? Word.
Exactly. Replace it with what? So what you will not find in this issue is one solution that magically replaces. But instead, 43 crazy ideas that nobody but a statistician can read. And in fact, most statisticians probably can't even make sense of those, right? So game over, man, game over. I don't know if you're familiar with Aliens 2, but I think it won't make any difference. It's probably the correct assessment there. So seriously, really, what do? Change the academic culture. That's what the industry would like. Good, please, give me a break. All right. Audit the labs, that's never going to happen. Come on. I mean, NIH is probably struggling to keep the budget it's got, right? I mean, I don't know how many years the federal government has been on continuing resolution over the past 10 years or so, but it's got to be almost all of them. There's not like extra budget going to come in for auditing. We don't get extra budget in our department to do additional assessment. We're just supposed to do it on the weekend, I guess. Right. Academics don't know what the value of an hour is in terms of dollars. Nobody's computed their hourly rate. You have to work for a living to do that. So are we going to give up p-values? So for what, right? I mean, one of the potentially more possible things is to write papers in terms of confidence intervals on effect sizes with graphs and things like that. That might actually be a more palatable option. There is a journal of applied psychology that actually banned the use of null hypothesis statistical testing maybe 3 years ago. And one of the papers in that volume of statisticians is, we looked at this journal and they're getting worse. But we still advocate banning p-values. So they did what we wanted to do, and it sucks. I don't know. Now, the guy who thinks all research is wasted in every discipline actually said, but if you divide p by 10, then that may work. So reduce p by a factor of 10, and everybody's going to chase this lower threshold for a while. I don't know. Is that going to change anything? Okay. Bayesian analysis. Well, that's too hard. Okay. I don't think we're going to do that. Some other oddball statistical approach. No, we're not going to do that. What do we do? All right. What's that?
Just like throw a PhD statistician in there.
Well, so in the UK, it is required to have a statistician on the team if you're doing animal research. In the US, it's not. And in fact, statisticians are almost never consulted unless a disaster has occurred and we can't get a publishable paper because we didn't get a small enough p-value. Okay. So one of the things that might help is at least if people could design their experiments in a way that this was a little bit more successful in the traditional paradigm, maybe that would at least help some. If you could get people to blind and randomize and understand something scientifically about their effect size, you might be able to get somewhere in the current paradigm. Okay? So, I mean, the explosion of software has led to a lot of the use of statistics without statisticians. Things like Graphpad Prism, which I had never heard of before I started talking to all these primate researchers and mouse researchers who were using it, I mean, those of us who tend to use programming languages like R think down on SPSS as this menu-driven thing, but Graphpad Prism is to SPSS what like a text editor is to Microsoft Word. It's a much more simplified thing. It's just barely different from Excel. But you can tell if somebody's using it when you see the graphs in their paper. I mean, it has a very distinctive kind of graph output. But so there is freeware to do this stuff, but it's generally for an expert to use. And in fact, what the NIH set us to do was to try to develop a piece of software that was maybe a more user-friendly statistical experimental design thing. And so that's sort of where we went. So you can sort of think here in the end, when I say the software that we wrote is the best, that you sat through all this for a commercial, right? Be sure to drink your Ovaltine, but I'm not going to. I won't do that. Okay, my colleagues will be unhappy with me for not selling. So let me say two final thoughts here. We found that almost everybody in the biomedical business took exactly one statistics class. Very few of them took an advanced statistics class. And they felt the pain of that, and most of them had learned the statistics they needed to know on the street, as they say, right? So, in this sense, I've become born again after doing this, and it's changed the way, and my students would perhaps think they're suffering for this, it's changed the way I teach our Math 204, that's an Introduction to Statistics class. Now, another thing is that the majority of the people we talk to are in biomedicine don't have any sort of sense of the statistical literature. In fact, when you talk to them in their office, they'll get out of statistics books like 50 years old. So this is the book that my advisor's grand advisor passed down through the generations for us to use. And it's like Fisher's teacher is the one that wrote this thing, you know. And they're somehow still finding a way to use this. Holy cow. Why would they search the literature and read these papers that are even unreadable to experts in statistics? I mean, they're busy keeping up with their own research, which is moving apace. So it's incumbent upon statisticians not only to think about how we're teaching the classes, but also how to reach out to people who are actually out there doing the business. So with that, I'm going to shut up, re-acknowledge my co-authors, Drs. Cousteau and Wong, the people who talk to us. I mean, there are some people who I think arguably could be accused of confessing their scientific frauds to us. We did have a couple of people actually say, I feel like you're my therapist after this. So NIDA paid us some money to do some of this research, and some of my traveling around was funded by the Wallen Endowment Fund. And over the past 3 or 4 years, a lot of Math 204 students have suffered through my attempts to try to atone for statistical crimes. So with that, I'm done.
Approximately 10 minutes for Q&A if anybody else has any questions. And we did begin tonight's program a little bit late, so because of that, the bar is going to be open until about 7:10. So before we go to the mingling, does anybody have any questions for Dr. Fitzpatrick?
Stunned him in silence. Carl.
But, I mean, you've alluded to...
Just because we're recording this.
You've alluded to that one of the maybe big changes that need to happen is kind of major shifts in cultures. Mm-hmm. And it seems, I'm not going to impart my own opinion on that. What's your outlook for the possibility of academic culture changing, NIH culture changing, the culture of how they fund research, how research is, valued? For instance, like, are you going to actually publish the experimental design? Put all that time into experimental design. Those changes have really significant effects on, output, productivity, time to translational research, and all those things. Based upon the work that you've done, what's your prognosis for where we're going?
Yeah, I have to say it's pretty scary. I think it's, once you've sort of started sailing in a certain direction, it's tough to change. I mean, in the time I've been at LMU, I've watched the sort of culture towards research and away from what had been almost entirely a teaching mission move, right? And at the large research institutions, I can't help but think that the pressure is going to continue to have more and more and more publications. I mean, that said, I mean, there are journals like PeerJ and PLOS One that will publish negative, quote unquote, negative results, by which I mean they didn't get a p-value of 0.05. It doesn't mean there's nothing there. It just means, you know, there's not as much evidence, perhaps. And I mean, that's a place where I could see a lot of potential support. If you, I mean, I would much rather see an honest study that came out and said our p-value was 5.6%, instead of all this dinkering around to try to pull it down to 4.7%. Because, like, once you've told me that many lies, how do I believe anything you've done, right? If you just be honest with it up front, that would be much better. I mean, that's, I think, a place where you could change things without a lot of grief. If journals would accept papers that are well-designed research, you know, the scientific premise is solid, and there's good scientific judgment that doesn't necessarily depend on this kind of artificial blame it on the statisticians cutoff, that would be a big step forward. And now, I mean, there's no real reason for page limitations in the same way. I mean, you might want, you know, sort of the short Reader's Digest version for Science Magazine, but also give credit for publishing the fuller set of stuff in the supplemental. I mean, I think if those things sort of both counted, then that would help with, you know, people having to get tenure and get promotion and get salary increases and get grants. So you could sort of look at the full productivity as inclusive of those additional activities. I mean, that's the only thing I can really think of with respect to academic culture. It's tough. It's a really tough problem.
So how does a layperson or a reader or a consumer of this information know what is valid and trustworthy, really, if even peer-reviewed journals aren't a safe source? I mean, we have librarians that did an article on peer-reviewed journals or journals, predatory journals, how to evaluate predatory journals and identify them, and how to evaluate peer-reviewed journals that are viewed as like our gold standard. And I just want to say that when I went to UCLA, I had a statistics professor who told me that you can manipulate the numbers any way you need to if you really understand statistics, which was initially a little frightening to me, and now you've raised that fear again.
Be afraid. Be very afraid. Another movie reference for you.
And I kept thinking of a similar thing too. Sorry not to interrupt, but like not just a layperson, but even as a librarian, here I am at LMU, supposed to teach the students like how to like rely on like credible peer-reviewed journals. How much more careful do I need to be? Could I just take a journal at its word if it just says, yeah, sure, it's peer-reviewed? Can I just sign off and say, like, okay, it's credible? How much more careful do I need to be now?
It's not clear how to solve this problem for consumers. And, I mean, one of the points that John Oliver makes in his Daily Show rant, or I guess his own show rant, is, you know, it seems like every week you see some study contradict previous study. And, I mean, it would help if journalists were actually a little bit more honest about what they were reporting on when you see this stuff in the news media, right? I mean, if you see, I mean, they'll say, oh, we did this study and blah, blah, blah happened. Well, it was really blah, blah, blah with a bunch of Sprague-Dawley rats. So why are you trying to necessarily jump to humans right away, right? I mean, everybody gets excited. Oh, there's this thing that's going to, you know, be a new treatment for cancer, this particular kind of cancer. Well, they did some testing in mice. And it turns out they did testing in this very particular kind of mouse that was genetically engineered to spontaneously develop kidney tumors. Which is, I mean, in order to sort of get something, a basic idea of whether or not it's going to work, you start with these kind of models where the occurrence of the thing is much more frequent than it would be the background in human beings. If we didn't just jump on the bandwagon of, holy cow, we got this new discovery so excitedly, I think that would help some. But this is a big problem. I don't think we have a good way of assessing this.
So, my favorite example just comes from a couple, like last week on NPR. Average allowance in the United States is $30 a week. No reference. So I emailed them in a fit of avoiding my other work and said, where did you get this from? And a few days later, they sent me this study, which then I was able to find eventually. And they surveyed 1,002 adults in the U.S., only 273 of which had children under the age of 25 living at home. And that they adjusted for age, gender, and race, but did not adjust for household income. So it was a study where they got about 275 people who had kids of unspecified age that had no idea what their income was, and they came to the conclusion that U.S. children have allowances of $30 a week. But I would say to the librarians that, yes, you would trust a peer-reviewed journal more than you would trust a predatory journal, but the buyer beware. And so that example that I kind of shouted out, that in Journal Club, we looked at a paper that had a migraine medication that changed the effective time of fixing the headache by about 15 minutes. And we discussed whether it was worth paying all this money for a drug that gave you relief on average 15 minutes sooner. And so as a consumer, you might decide whether it's worth it to you or not worth it to you, and whether your insurance company pays for it or not. But we have to have some sense of basic critical thinking and nuancy and questioning it, and the idea that just because it's published in a peer-reviewed journal does not make it true. But I wanted to ask Ben, on a different note, where does data sharing fit into all of this? So, the other part of a lot of discussion going on in the scientific world right now is about data sharing. And so in the fields of bioinformatics, there's a lot of folks that have been on the data sharing bandwagon and open source software and open access publishing for a long time. And then there's a lot of people that are resistant to it, in which I would include people that maybe get funded by NIDA, that don't want to share their primary data for various and sundry reasons, and think of data scientists as parasites that are mining their data and they should have access to it. And so do you think that improving data sharing would shine a bright light on some of this in a way that might be hidden in dark corners, because people aren't usually required to share the primary data? They usually share like summary statistics.
I think sharing the primary data would be a big step forward, and with the caveat that you actually have to make it readable and usable. Right? I mean, people will send you a data, yeah, sure, I'll send you a data set, and they send you some Excel thing that's got no labels, and how do you figure this out, right? And, I mean, this is something that NIH could very well require, right? I mean, they pay for the data. They should get it. And they should build database. I mean, there are government organizations that are trying to build databases. Usually it's focused on a very specific thing, like addiction or traumatic brain injury or something like that. But we got a long way to go on those things. And it seems like we're largely focused on these big things like functional MRI type data or microarray data. These, genome-wide association studies have vast amounts of data. And an animal study that has maybe 50, 60 data points and a couple of hundred numbers in it somehow doesn't seem quite as important, but in fact it is important. The flip side is there are a lot of people who are very concerned about getting scooped, right? We generate this data, we haven't been able to figure it out. If we go to those guys, they might find what we're not finding. I mean, that's an issue. So, I mean, but at the bare bones minimum, when your grant runs out, you ought to have to turn it over to the NIH. I mean, that would be my feeling about it, yeah. And I think it would help.
And if I could just ask, on your repeatability scale, from repeatability to reproducibility, like I've read some people discussing and saying, or to generalizability, that we really shouldn't be worrying too much about repeatability, and that really what people are worried about is whether this answer is going to be generalizable to another system. And so I guess I want to ask is, would you even be able to get to generalizability if you don't have repeatability in the first place?
My feeling is we can't really worry too much about that until we do get a handle on these sort of basic repeatability things. I mean, somebody in the same lab ought to be able to redo the experiment and get the same thing. And labs that have strong training components, and there are many, you undergo these kind of boot camp type processes where the new kids come in and they have to first repeat the old experiments. Now, that's expensive, and so it has to be a lab that has a decent budget to be able to tolerate that kind of stuff. But if you can't do the most basic repeatability things, then how are you going to do the generalizability, right? I mean, the same thing's true for me when I write codes. If I can't do these very simple test cases, how am I going to take this code and use it on a complex problem? I believe you had a question.
So for the people you talked to, you interviewed and so forth, I guess I'm thinking about the sort of the relevance difference in p-values. So if someone does a study and p-value is 0.56 and they manipulate the cases to get it 0.5 or below so they can publish it, I guess, and then they're saying that that's they couldn't reproduce those or replicate those results, right? But when they did try to replicate, if they were getting results of 0.58 or 0.6 and saying, oh, we can't reproduce that, I mean, really, that's a pretty insignificant difference to, you see what I'm saying, drawing the threshold. When they were trying to replicate the studies or get reproduced results, if they were drawing a hard line at, say, p.05, the percentage of, if they had drawn a line at p.06, that the percentage of replicated studies would be a lot higher.
So maybe not. I think part of the issue is that people are chasing a threshold, and as soon as you draw the threshold, then people are sort of manipulating the studies to hit whatever the threshold is. And so what gets published, whether it was meeting this threshold or that threshold, we got a bunch of papers that are sort of, the distribution of p-values in published work is not some kind of well spread out thing. People have done these kind of meta-studies where they've looked at p-values of published work, and you find that there is a kind of a remarkable concentration, in the 0.045 to 0.05 range, where people are clearly chasing that threshold of success. Right. So, I think, the medical doctors who are suggesting decrease it by a factor of 10 will eventually find the same phenomenon happening at that much more restrictive threshold. That if I really reduce my tolerance for false positives by that much, eventually you're going to find, the number of subjects in the studies, whether it's survey respondents in a social science research or animals or patients in a human study, the numbers will have to go up in order to get that sort of finer data point, but you're still going to have similar problems. I think what statisticians are recommending that's maybe a little bit hard to implement because it's kind of fuzzy is report all the values and make your assessment on multiple things, not just the p-value. You will find papers where they don't even tell you what the number is. They'll say, for this thing, p was less than 0.05. For this thing, p was less than 0.01. Social science papers often have asterisks, right? And one asterisk means 5%, 2 asterisks means 1%, 3 asterisks means 0.1%. and they don't even report the numbers. At least report the numbers, and in fact, report what you estimated and what the wiggle room in that estimate is. That would be a much better way to report results.
This might be the last question of the evening. Make it a good.
Hi there. So I'm coming from a social science background, so I can't speak as much to the pharma or biomedical side, but I was just wondering what your thoughts are on the idea that in addition to the problem of bright lines, that there's not as much emphasis as there should be on statistics being about uncertainty, especially in the context of social science. And like you said before, there's this